Kiona Elliott. I'm a graduate student in Becky Bart's lab at the Danforth Center. And today I'm excited to share with you all some of the work in our lab looking at genetic and epigenetic strategies to reduce cassava susceptibility to bacterial blight. And a lot of the work that I'm going to be sharing today has been done by a lot of people in the lab, so I'll be sure to let you know who's done what as I go throughout the presentation. So the disease that we're interested in, one of the diseases that we're interested in the lab is cassava bacterial blight. It's the leading bacterial disease for cassava, and it's caused by the pathogen Xanthomonas ixonopodes pathovar manihottis, or XAM. You can see an example of what cassava bacterial blight looks like in the field. Here's an image of a field site that's been impacted with CBB in Nigeria, which was sent to us by one of our collaborators. And some of the disease symptoms for CBB include these water-soaked lesions that appear on the leaf. They're one of the early indicators of cassava bacterial blight. So they're these dark, wet spots that appear at the site of infection, along with leaf wilting, stem browning, and eventually death of the plant. One of the ways that XAM is able to contribute to this disease is through the use of transcription activator-like or TAL effectors. These are proteins that the pathogen is able to secrete into the plant cell through the type 3 secretion system, and they can localize into the plant cell nucleus where they bind to a region known as the effector binding element, or EBE site. And this EBE site sits upstream of susceptibility, or S genes, in the host plant. And so when a TAL effector binds to the EBE site of an S gene, it leads to either the ectopic or overexpression of that susceptibility gene, and this helps to promote pathogen virulence in the plant in some way. One of the S gene targets that we are interested in the lab is meswit, which is a part of a family of sucrose transporters. It is ectopically expressed by the TAL effector TAL20. So when TAL20 binds to the meswit EBE site, it leads to sucrose being pumped out into the extracellular space where the pathogen is proliferating. And from prior work, we know that if we were to take XAM and make a mutant where it no longer can make the TAL20 effector, it causes a reduction in the water-soaking phenotype that I described earlier. So with this knowledge, we have this hypothesis that if we were to prevent meswit induction by the TAL20 effector, then we can reduce cassava susceptibility to the disease. And we wanted to have a really nice way of being able to measure cassava bacterial blight disease severity. And so we took advantage of water soaking as a phenotype and developed an assay where we take cassava plants, we infiltrate them with XAM, the mutant that does not have TAL20 or mock treatments. And then at days 0, 4, 6, and 9, we detach that infected leaf and image the leaves using a Raspberry Pi camera system we have set up in the lab. And then we can use these images for image analysis to extract quantifiable information about what that lesion looks like. So a big part of my thesis work has been developing 2 different methods for analyzing water soaking and tying that to CBB disease severity. The first is an ImageJ method where we take each of our images and we generate a mask for every single image. So we outline the water soaking spots manually, threshold and convert it to a mask, and then use that mask and overlay it to the original image to extract information about the lesion. And from this data, and wild-type plants infiltrated with the wild-type XAM mock treatments or mutant treatments, we see that when we look at the total water-soaked area of our lesions, the wild-type XAM plants have a larger lesion area, or the wild-type XAM inoculated spots have a larger lesion area compared to the TAL20 inoculated sites. And we're also interested in the color of these lesions because when you look at the lesions by eye, we notice that the TAL20 lesions appear to be a little bit lighter. And when we look at the data for that, we see that the grayscale, which is our measure of the lightness or darkness of that lesion, for wild-type XAM inoculated spots are darker compared to TAL20 or mock inoculated spots. So really excited that this data was able to allow us to separate out between our different treatment types and that we could use it as an indicator of disease severity. However, using ImageJ is a pretty time-consuming method because, again, you have to make a mask for every image. And so we wanted to develop a quicker way of doing this. And I worked with a data scientist at the Danforth Center, Jeff Berry, to develop a machine learning-based tool for still getting that information about the lesion. I think someone's not muted. Kanan, I think I can just mute you. There you go.
Oh my.
Okay.
Oh, is it okay now? I didn't hear any.
Yeah, I got it. Okay, so for this machine learning based tool, we develop a classifier file using representative images of infected leaves at different time points, and we can create a single mask from there and a file that we can then apply to all of our images and process them in order to get that information. So again, looking at our data set with this tool, we first looked at water soaked area. And again, we see that the wild type SAM spots have a higher, significantly larger area compared to Tau20 spots. And again, we also see that for the grayscale mean, the lesions are darker for the wild type SAM spots as well. And both of these different methods that I've shared with you, we recently submitted in a paper, and this work was done again with my lab mate, Jeff Berry, as well as a high school intern who I trained on the ImageJ analysis method. So to summarize as part of my talk, what we've been able to show both of these both the machine learning and ImageJ image analysis methods are that we're able to quantify water soaking and then use this to kind of score our cassava bacterial blight disease severity. And so next I'll move into how we're applying this to our overall goal of modifying the me-sweet-10A gene or EBE site in order to reduce cassava susceptibility. We have two different strategies that we're using to go about developing these less susceptible lines. The first is using CRISPR-Cas9 to either prevent the S gene activity, remove the EBE site, or edit it enough so the TAL effector can't bind. And the second is an epigenetic strategy that's led by my lab mate, Kiona Elliott, where she is methylating the EBE site in order to prevent TAL effector binding. So first I'll show you some of the results from our CRISPR-Cas9 work, where we're using a dual guide RNA strategy to target the Tau20 EBE and right after the translation start site. And from these guide RNAs, we have two mutants that we've moved forward with, a frameshift mutant where the EBE is intact, but there's a premature stop codon in the first exon of me-sweet-10A. And the second is a deletion mutant where the EBE, the 5' UTR, and the translation start site have been removed. And when we look at these plants after they've been inoculated, we see that they appear to have a smaller lesion and lighter color compared to wild type plants. So we applied the machine learning tool to this to quantify these differences that we're seeing by eye. And we can see that both of the mutants have a decreased water soaking area compared to wild type plants when infected with SAM. And when we look at the grayscale, we can see that the lesion color is significantly lighter compared to wild type SAM. And so this data was a really nice proof of concept to show that if we were to prevent me-sweet-10A induction, then we're able to reduce disease severity. And so this made us more excited for moving forward with the epigenetic modification work that's led by Kira. And so this work that I'll be telling you about has recently become available on bioRxiv, so I encourage you all to check it out. And I'll remind you that the hypothesis that Kira is working with here is that if you were to methylate the EBE site, it would prevent the TAL effector from binding. And the tool that she has developed is a zinc finger that targets me-sweet-10A, and it's fused to DMS3, which is a component of RNA-directed DNA methylation pathway from Arabidopsis. And this DMS3 component in this pathway has been really well studied by our collaborator, Steve Jacobson, in Arabidopsis, and he's a collaborator for this project as well. Okay, so first I'll show you how Kira has developed two different lines using this tool. So we'll be looking at some bisulfite sequencing data to look at methylation in two different lines that she generated. And just to orient you, we'll be looking at the me-sweet-10A 5' UTR area, specifically the EBE, which will be highlighted with this gray box. And along this axis, we're looking at the amount of methylation. So when we look at the bisulfite sequencing data, we have two different lines that were generated, 204 and 133. And we look at the EBE site, we see that these two lines have methylation peaks that are within the EBE site, but this methylation is absent in our wild type control. So next, Kiera was interested in whether or not these methylated lines have reduced induction of MeSWT when they're infected with SAM. So she worked with our lab mate Greg in order to test this out, where we took SAM infected leaves and after 2 days collected them for RNA and qPCR. And what they found is that both the methylated lines 204 and 133 have reduced induction of MeSWT after induction with SAM. And here is an example of what one of those lines look like, and it looks very similar to the wild type plant morphologically. So the next question that we were interested in is, are these methylated lines actually less susceptible to cassava bacterial blight? So we applied the water soaking analysis method here as well, and infected the plants with SAM and the Tau20 mutant. And we could see that the water soaking was significantly decreased compared to the wild type inoculated plants. And it looked more similar to the Tau20 mutant inoculated sites in wild type plants. So when we apply the tool to this and look at it quantifiably, we see that both of the methylated lines have less water soaked area compared to the wild type plants, and the water soaking area between SAM wild type and Tau20 in both methylated lines is more similar to each other compared to the difference that we see between the two in the wild type plant. And we also see that these mutants have decreased or lighter lesion color as well compared to the wild type plant. So really excited about these results. And again, this data is available in a preprint that's on bioRxiv now. I'd like to wrap up my talk with a couple of take-home messages. So first, we have these two nice methods that we're using to measure cassava bacterial blight severity. And using this method, we've been able to show that two MeSWT lines have decreased severity. We've also shown that the DMS3 zinc finger technology is able to methylate the Tau20 binding site, and that this methylation prevents Tau20 binding and activation of MeSWT. And also using the image analysis method, we see that the methylated lines have decreased disease severity as well. And so with that, I'd like to say thank you all for listening, and thank you to all of my lab mates, especially Kiera, for all of her work with leading the Epigenetics project. Jeff and Hoban, who worked with me a lot for the image analysis method, our collaborators, and our funding sources, the Bill and Melinda Gates Foundation, NSF, and others. I'll take any questions. Thanks.